What's up, everybody? It's my big T. Merry Christmas. Merry Christmas. Anyways, Big D, Talent Talks, biology of belief. I'm actually pretty excited that the last episode I did got 11 views, 11 plays. Good job. Yeah, we're actually starting to motor through this book pretty good. So let's finish the last little bit that we were talking about. We were talking about The Secret of Life was the section that we were reading. And continuing on, I'd been trained as a nucleus-centered biologist as surely as Copernicus had been trained as an Earth-centered astronomer. So it was with a jolt that I realized that the gene-containing nucleus does not program the cell. Environmental data is entered into the cell/computer via the membrane's receptors, which represent the cell's keyboard. Receptors trigger the membrane's effector proteins, which act as the cell's computer's central processing unit, CPU. The function of the computer CPU is to convert incoming data into the binary code language used by the computer's operating system. 1101001101. Computer code operating ones and zeros. I guess until you get into quantum computing and then it changes. But we're not going to discuss that. Not this time. I don't know enough about it. The receptor effector protein complexes represent a functional complement of a computer CPU processor. Incoming environmental information is passed from the receptor to the effector protein, which in turn converts the incoming signal into the behavioral language of biology. I realized in those early morning hours that even though biological thought at the time was still preoccupied with genetic determinism, leading-edge cell research, which continues to unfold the mystery of magical membrane in ever more complex detail, tells a far different story. At the moment of transformation, I was frustrated because there was no one with whom I could share my excitement. There's been times where I have had realizations, and in those moments, I just call someone. And I have to be like, yo, I just had this crazy realization. Rah, my mind's blown. You know what I'm saying? So do that too. Do that. Call. I was alone out in the country. My house didn't have a telephone because I was teaching at a medical school. I realized that there was undoubtedly be some students studying in the library. I hastily threw some clothes on and raced off to the school to tell anyone of this exciting new insight. Running into the library, out of breath, wild-eyed and my hair flying in all directions, I was the epitome of the absent-minded professor. I spotted one of my first-year medical students and ran up to him proclaiming, You have to hear this. This is a great... pardon my language, he wrote it in the book. Okay, so he's like, you have to hear this. This is great shit. Yes, you have to hear this. That reminds me of Doc from Back to the Future. Marty, you've got to come quick. Anyways, I remember in the back of my mind how he pulled away from me almost in fear of this raving mad scientist who wildly broke the silence of the sleepy library. I immediately began to spew forth my new understanding of the cell using the complex polysyllabic jargon of a conventional cell biologist. When I finished my explanation and was silent, I was waiting to hear his congratulations, or at least a bravo, but nothing was forthcoming. He was now wide-eyed himself, and all he could say was, Are you okay, Dr. Lipton? I was crushed. The student had not understood a word I had said. In hindsight, I realized that as a first-semester medical student, he did not have enough scientific background or vocabulary to make any sense out of my apparent rantings. However, the wind was knocked out of my sails. I held the key to the secret of life, and there was no one who could understand me. I confess I didn't have much better luck with most of my colleagues who had been schooled in syllabic jargon. So much for the magical membrane. Over the years, I gradually honed my presentation about the magical membrane and continued to refine it so that first-year medical students and lay people can understand it. I've also continued to update it with the latest research. In doing so, I've found much more receptive audiences among a wider range of people. I have also found audiences receptive to the spiritual implications of my eureka moment. Shifting to the membrane-centered biology was exciting for me, but it wouldn't have been enough to send me screaming to the library. That Caribbean moment not only transformed me into a membrane-centered biologist, it also transformed me from an agnostic scientist into a card-carrying mystic who believes that eternal life transcends the body. I'll get to the spiritual part of the story in the epilogue. For the moment, let me reiterate the lessons of the magical membrane, which put the control of our lives not in the genetic roll of the dice at conception, but in our own hands. Right there. Put the control of our lives not in the genetic roll of the dice at conception, but in our own hands. We are the drivers of our own biology, just as I am the driver of this word processing program. We have the ability to edit the data we enter into our biocomputers, just as surely as I can choose the words I type. When we understand how imps control biology, we become masters of our fate, not victims of our genes. I can't say that mainstream scientists have taken on my spelling of membrane, and neither that they have been tooting my message that integral membrane proteins make us masters of our fate, but I could say that there is a wealth of new and fully supportive research about how the membrane interacts with the environment to shape biology. For example, research on the cell's membrane potential, which I talked about above, has opened up a new way of thinking for developmental biologists who had previously only focused on the role of single molecules, hormones, neurotransmitters, or other chemical agents, in controlling development and creating body parts. In 2011, biologist Michael Levin's team at the Tufts Center for Regenerative Why can't I say it? Regenerative. There we go. Regenerative. And developmental biology altered the bioelectrical voltage of the membranes of tadpole cells. Amazingly, simply altering the membrane potential in the cells from the backs and tails of tadpoles resulted in fully developed eyes growing in the backs and tails. Ooh, eyes growing in the tails? Far from where eyes normally form. Sounds like impressive magical membrane to me. The key to success of the study was the team's findings that during the embryonic development of a tadpole, the membrane potential in cells destined to form an eye drops precipitously from about 70 millivolts to about 20 millivolts. In their lab, Levin's group induced the same drop to 20 millivolts by inserting voltage-regulating calcium ion channels proteins into the membranes of the tadpoles. Just so you know, I have no idea what any of that means. But, right on. Channel into the membranes of the tadpole's back and tail cells, triggering a signal that initiated the growth of a complete eye. This research is exciting because it opens a possibility of repairing birth defects and regenerating damaged human organs. It also underscores the fact that the membrane controls cell behavior using non-chemical electric environmental signals. Aside from the regenerative medicine applications of this new technique for eyes, this is the first step to cracking the bioelectric code, said Levin. Membrane research is also helping rehabilitate the reputation of cholesterol, which has long been vilified as a culprit in the modern-day scourges of cardiac disease, heart attacks, and stroke. In a case of guilt by association, high levels of cholesterol are present in 35% of patients with cardiovascular disease, and at sites of vascular damage, the endothelial cells, the inner lining of blood vessels, are loaded with cholesterol droplets. But I want to offer a more nuanced view of cholesterol, which is often lost in the rush to demonize it. Cholesterol is a lipid molecule that plays a vital role in our day-to-day survival. It is, for example, the precursor for the synthesis of important steroid molecules, including the bile salts used in digestion, regulatory steroid hormones such as estrogen and cortisol, and vitamin D. More relevant to this chapter, cholesterol is an essential component of the membrane whose function is required for the survival of our 50 trillion cells, which is another way of saying our survival. Cholesterol helps the membrane maintain a very important balancing act. It must be rigid enough to physically resist the strain placed on it by the cytoplasm it encloses, yet supple enough to accommodate the flexibility required for movement of cells. Membrane fluidity is also of great importance in controlling the cell's brain function because it impacts the membrane's ability to read and respond to environmental information. To function normally, IMPs in the form of receptor and effector molecules must be able to engage one another by freely circulating within the membrane's inner, oil-loving, hydrophobic core. It is the viscosity of the membrane's lipid core that controls the ability of these proteins to circulate freely. A membrane made up of only phospholipid molecules would be quite fluid, so it would enhance the mobility of IMPs, but it would not be rigid enough to hold up under the pressure generated by the enclosed cytoplasm. Cholesterol is a more rigid molecule than phospholipids. So when cholesterol is inserted into the membrane, it mobilizes surrounding phospholipid molecules, creating the extra rigidity that strengthens the membrane and impedes the flow of small ions and molecules into the cell. The inserted cholesterol also creates extra space among the phospholipid molecules, space that keeps them from gelling so that the phospholipid molecules don't turn from an oil-like substance into a butter-like substance. So in addition to stiffening the membrane, cholesterol acts like antifreeze. That ensures that proteins and lipids are able to move freely. Counterintuitively, the membrane's rigid cholesterol molecules can act to restrict IMP movement. When clusters of cholesterol molecules link up with a class of lipids called sphingolipids, they form structurally rigid rafts that restrict the movement of entrapped IMPs. This restriction on IMP movement offers another example of the power of banding together for the greater good. The rafts behave like corrals that group clusters of IMPs so they can work together to control specific cellular functions. Cholesterol rafts are the cell's equivalent of short-term memory because the IMPs they contain represent information that engages a variety of cell behaviors. These vital roles suggest that cholesterol should not be considered a feared evil villain, but instead just a foot soldier doing its duty somewhere down the line of command. I was never predisposed to convicting cholesterol for causing heart disease, because when I was transitioning from the laid-back life of a Caribbean lecturer to the far more frenetic life of a scientist at research juggernaut Stanford, I spent time at what I describe jokingly as research halfway house at Pennsylvania State University in the lab of Theodore M. Hollis, a gifted scientist I met when he guest lectured at the Island Med School. When I was in his lab, Ted showed me samples of blood from the specialized strains of rats he used to study human atherosclerosis, the hardening and narrowing of arteries that is the leading cause of death in the United States. These animals had so much cholesterol in their systems that their blood was milky white. Despite their apparently toxic level of cholesterol, these rats did not form endothelial cell plaques typical of atherosclerotic. I'm just butchering words in this book today. Blood vessels. The secret Ted added an over-the-counter antihistamine drug, the same kind that allergy sufferers turn to regularly. When he introduced the cholesterol, because the antihistamine could override the cholesterol's apparent role in atherosclerotic plaque formation, his work showed that the mere presence of the cholesterol was not the driving force behind a blood vessel's malfunction. Since antihistamines protected the rats, Ted's research obviously suggested an alternative culprit: histamine. Note, despite my friend Ted's exciting research on rats, I am not advocating that humans load up on antihistamines. The research is too preliminary for that, and as you know, I think that all too often, biomedicine rushes to drug solutions without fully understanding their side effects. Histamine is a stress-related hormone that prepares the body to deal with anticipated injuries and inflammation, when the fight-or-flight response is activated by a perceived stressor. Now, decades later, the role of histamine in facilitating atherosclerosis has been confirmed. In recent mice studies, the gene for histamine synthesis were experimentally knocked out. These genetically modified mice, unable to synthesize histamine, resisted the influence of stressors that led to inflammation and atherosclerosis in control mice. And the protective results observed in histamine-free mice were independent of serum cholesterol levels. The results of animal studies point to the role that chronic stress plays in the creation of histamine and in the onset and exacerbation of atherosclerosis and promotion of cardiovascular disease. In direct contrast to the implied role of cholesterol in causing heart disease, cardiovascular pathology may instead primarily result from environmental stressors rather than genetic or biochemical dysfunctions. Though this research argues against the medical establishment's rush to judgment against cholesterol, that rush was fueled by the interests of the pharmaceutical industry, of course. That's because the drug companies had come up with another one of their beloved magic bullets, this time in the form of statins. Statins are a class of drugs used to lower levels of cholesterol in the blood by inhibiting a liver enzyme responsible for producing 70% of the body's cholesterol. Statin drugs were originally intended for high-risk cardiac patients, but someone, likely in sales, came up with the idea that statins might be good for primary prevention to help those at risk of developing heart disease in the future as well. The Jupiter study, frequently referenced in support of statin use, found that during the study period, there were 68 heart attacks in the placebo group and only 31 heart attacks in the group that took statins. So according to those numbers, statins produced an astonishing 58% reduction in relative risk. The results led by the research group to advise the statins were effective for primary prevention of heart attacks. On the surface, those stats sound very impressive, but the suggestive conclusion was simply a manipulation of the data. It should be noted that the experimental and control study groups each had 8,901 participants. In real terms, the heart attack risk went from a very low 0.76%, 68 out of the 8,901 in the control group, and 0.35%, 31 out of the 8,901 in the statin group. Statistically, the protective effect of statins provided for a 0.35% reduction over controls, which meant a real risk reduction of less than 0.5%. The data indicated that for every 300 people taking expensive statin drugs, only one life might be saved. Holy smack. Follow-up studies reveal that the presumed preventative effects of cholesterol-lowering drugs have been considerably exaggerated. As a side note, AstraZeneca, makers of the statin drugs used in the study, was the source of funding for the now discredited Jupiter study. The use of statins in the primary prevention of heart disease has fueled statin sales, but it hasn't turned the tide in the war on cardiovascular disease. In fact, as with many wars waged lately, the cost is high and the results negligible. Though statins accounted for $29 billion in U.S. sales in 2013 alone, their war against cholesterol has barely had an impact on cardiovascular disease. At best, statin drugs lower the actual risk of heart attack by around 0.3%, while at the same time producing side effects in 15% to 40% of those using the drug. Recent independent studies have shown that statin use for primary prevention has minimal or no value in reducing heart attacks and mortality. The statin approach to treating cardiac disease is yet another expensive war with a very poor prognosis. As of yet, we haven't found the weapons of mass cardiac destruction. Instead of continuing to search, perhaps it's time, actually way past time, to revise the conclusion that cholesterol is culpable for cardiovascular health issues and shift our attention to environmental stressors rather than genetic or biochemical dysfunctions. So there's a little bit of information on statins and cholesterol. Who was it? There's this lady I follow online, and she talks about cholesterol, how cholesterol used to be fine at 350 and now they want it below 150. I don't know. I haven't done any research on cholesterol. I don't know anything. I'm just saying that from what she was saying, that cholesterol is used for a lot of functions within the body, especially the brain. Don't quote me because I don't know very much at all. So I'm not sure. If you know, comment. Give me some info. So yeah, there's some information on statins. The origin of 90% of cardiovascular disease is not due to an organic dysfunction in the cell's mechanisms, but rather represents a behavioral response driven by environmental signals in the blood. The brain secretes blood-borne hormones, stress factors, and inflammatory agents in order to coordinate the function of 50 trillion cells to sustain life. This insight returns us full circle to the story of the magical membrane, because the cell membrane is the information processor that provides the interface between biology and our brain's perception of the environment. A more complete understanding of cholesterol's vital role in membrane information processing makes it apparent that disturbing cholesterol metabolism with statin drugs is tantamount to throwing yet another monkey wrench into the machine. Until recently, disease was perceived as a consequence of breakdown in cells biochemical mechanisms. The vast majority of disease is now recognized to be the result of lifestyle. When biomedicine fully appreciates that the membrane is a truly magical brain, it'll shift away from genetic/biochemical dysfunctions and focus on how we can change our perceptions/beliefs, more in later chapters, to prevent cardiac and, in fact, almost all other diseases. Wow, we're on chapter 4, you guys. Chapter 4, the new physics. And this one's going to be called Planting Both Feet Firmly on Thin Air. So with that, we're going to end it. I'm really trying to get my narrating down so it's a lot better, or reading so it's a lot better. And in order to get better, you must practice. So that's why I read to you guys, because it's my practice. And we all learn. And we're revisiting a book I read a long time ago, probably a few years now. So revisiting it, it's a nice refresher. Oh, excuse me. Anyways, we're up to 52 countries. 52 countries tuning into the podcast, even if it's 0.1%. I'll take it. If there is a chance that I could just spark and ignite a flame within inside of all of you to work on this, to change this, it's all worth it. It's all worth it. And you get to laugh at me. Right? You get to laugh at me because I mispronounce words. And sometimes I make not so funny jokes. But at least you're laughing. Anyways, with that being said, thank you very much for tuning in. I appreciate every single one of you. I'm glad you joined me on this learning journey that we're on. And I'll catch you at the next one. Cheers.